everybody, I'm Kenzie. My project this summer was over the distribution of Asian clams in the St. Louis region. My advisor was Danielle Hake. In this map you can see our sites that we sampled. We went into Jersey County, mostly in Madison County, and then over into St. Louis County. So, the Asian clam. When did it come over to, first of all, it's an invasive species, and it came over to the Americas in the 1920s, likely via ballast waters, or as a food source from Asian immigrants. It has now been found in 85 of 114 counties in Missouri and 93 of 102 counties in Illinois. The first documented sighting of them was in the 1970s, but they were likely there before in the St. Louis area. They were likely there before that, so this gives us reason to believe that in our area they would have established populations. What is an invasive species? So invasive species are often detrimental to native flora and fauna. Specifically, the Asian clam is detrimental to the native mussels because it filter feeds at an incredibly high rate, and it filters out their food and their sperm, so they are not able to eat or reproduce. Invasive species can be costly and difficult to remove. Specifically with the Asian clams, they have tried bottom barriers and diver-assisted suction removal. That was found to be ineffective in Lake Tahoe. They also tried heat treatment, which they heat the sediment up past a temperature that the clams can live in, but that also failed in Lake Tahoe. And then at Lake George, they saw what had been happening in Lake Tahoe and were like, we don't want that to happen to us. So they, at the first sign of clams, started efforts to remove them, and they used the benthic barriers. They found that it was effective short term. They would come back so they do this in the fall, right before the reproductive season of the clams was over. And the following spring, they didn't see any, but eventually the clams would come back. So that was also ineffective. So there's no effective way to get rid of them, essentially. The objectives of our study were to find populations in the area and determine their population dynamics. Eventually, we went far enough into it to try and determine if they were a source population or a sink population. So once we got to a site, we would put out this rope, and the rope had multiple knots along it, and at 5 knots on the rope, we would put flags. And at each flag, we would place our little square PVC pipe thing. And we would dig 6 inches deep and then sieve through that sediment. First, using the green sediment, or sieve in that picture there. Sometimes, depending on the sediment type at each site, there would be tons of giant rocks in the green sieve, and then other times there'd be almost nothing. Once we got through with the green sieve and examined it for adult clams, we would move on to smaller sieves. You can see the gold one pictured there that are 2 millimeters. We would scoop the sediment out of the bucket that we caught it in after the green sieve, and we'd place it into the gold sieve, and we would look for clams in that one. If we found some in there, they were incredibly tiny. Once we thought that we had found a live clam, we placed them into white trays with water and tried to observe them moving. They don't like where they're at whenever you place them into this white tray, so they would immediately stick out their foot and try to roll around finding a suitable habitat. So if they didn't do that, within like the 5 to 10 minutes that we had them in the tray, we would determine them to be non-living. Once they were confirmed to be living, we would measure them with our calipers. And we measured the top to bottom, beak to umbo, and then also side to side. In the field, I would collect 6 ounces of water and 2 cups of sediment, and then once we returned back to the lab, we would filter out the water, and then I would analyze it for total dissolved carbon, total dissolved nitrogen, phosphate, nitrate, and ammonium. And then the sediment we would dry out. That could take anywhere from 2 days to, I think at one point I had to wait a week and a half for it to dry out completely. And then once it was completely dry, I was able to sieve it through 5 of these sieves at varying widths. And the picture on the top left is examples of full sets of sediment fractions. And the bottom one is what has been captured in a 2 millimeter sieve. And then here's what our results were. The highlighted ones are areas where we found live clams. The yellow is small shells, the gray is adult shells, and then the orange and blue are alive. So you can see that we didn't find many adult clams at all, and in general we didn't find many alive ones. So this is where we went from just doing a population survey to trying to determine whether they were source or sink populations. And here are our sites that those came from. You can see on the legend whether we found live ones there. None are shells. And you can see that where we found most of the live ones was over in St. Louis. And then here I have highlighted the same sites that had live clams so that we can look at their sediment makeup. And as you can see, a lot of them liked the gravel. So the dark blue is gravel. And a lot of them had high percentages of gravel, which this was contrary to what literature had shown before we had started the study. So this was a pretty important finding. Here, this is only sites that we had found signs of clams plotted against their percent of sediment type. The black is the gravel, the blue is the sand, and the red is the silt. We decided that the sand had marginal significance and that with more sample sites, we would likely see a higher p-value. And it was a negative correlation, which was opposite of what literature had said. And this is the almost same graph, but it has all sites included. So you can see a bunch of the zeros at the bottom. And here the silt was significant, but we believe that only to be because of how many points are plotted at 0, 0. The R squared wasn't very high. So in conclusion, you can see in the map on the left where we determined that there were likely sources, possible sources, unknowns, possible sinks, and likely sinks. So in a lot of the areas, we determined that we had just not found enough evidence of clams being there to determine whether or not it was a source or a sink population. And most of the source populations are in St. Louis. We do believe that the other source is the Mississippi for all the other ones along the Mississippi River, because we did go out and observe clams of adult size. And then the pictures on the right are of the site at Deer Creek, which was the one where we found the most adult clams at. And the bottom picture is pictures of discarded shells from likely raccoons or possums. And the top picture is what the sediment looks like. So we believe that they're choosing gravelly sediments with large rocks due to the raccoons or possums not being able to get to them underneath the large rocks. So we believe predation has a large influence on their sediment type choices. And here are my acknowledgements. And does anybody have any questions? Yes.
So where you did find the clams alive, did you find that, was it like patchy in the stream, or were they just kind of evenly distributed? And if it was patchy, where'd you find them?
So it really depended on the site. Like this one pictured, close to the stream, like the side of the stream, didn't have as many big rocks. So we believe that the raccoons were easily able to access the clams in that area. So mainly there we only found shells, and we were finding the live ones further in underneath larger rocks. But then in other sites it was pretty even across the board. Brooke?
Did you find any literature that assessed like the microhabitats of the clams in their native habitat?
No, I did not.
I was wondering.
Yes.
So how do they migrate upstream? It just seems that if they release feligers, or like the little baby mussels, that they'll float with the water downstream. How do they move upstream?
So that's how a lot of mussels move, is how you described, but we believe that during the flood events, they are carried upstream through the backwaters, and then they just keep on coming up.
What about this species allows it to outcompete the native species of clams that we're going to have around here?
Its filtration rate is incredibly higher than any other mussel that is in the water.
How about zebra mussels?
So zebra mussels everybody knows about because they cause direct issues to humans with the power plant blocking up their exit waters and stuff like that. I'm not exactly sure what their filtration rate is, but they're more of an issue because they stick together, and the Asian clams don't really do that.
And what did the water quality data end up meaning for the clams?
None of it was significant. It was pretty even across the board. So, yeah, all that worked for insignificant data. Sorry. Any other questions?